Okay, thank you very much. So I had the task today of really kind of touching on what it is that we're talking about when we talk about left versus right. I think this is an area where we have, over the past few years, just moved into the complexity of biomarkers, right? We talked about RAS, what codons do we test? Do we include NRAS? All these things that we've moved in really picking our patients. And all of a sudden this comes on the scene, that maybe it's just this simple left versus right. And so I think what the goal here was really to say, what is the underpinning? Why does this make sense? What is it that we're talking about when we use this very simple surrogacy of left and right? So this is biomarkers and biology. First, just as a reminder, that when we talk about right and left, we're really talking about parts of the colon that are fed by the superior mesenteric or the inferior mesenteric artery. These are what the surgeons tell us when they say we did a right hemicolectomy or left hemicolectomy, where they're following that vascular distribution. Classically, it is kind of the third, the most proximal one-third of the transverse is lumped in with the right. But as you know, the anatomy is certainly variable. And so we run into this problem of what's the transverse. And the reality is we don't know. We can't really, when we're doing a colonoscopy, we can't tell what artery is feeding it. So we have a little bit of a gray zone, but it really is a definition. You heard a little bit before about the prognosis of this, and I just have a few slides to reiterate this point. I love this study, the E2290. This is the 5FU era. And what we're plotting here are months, and these are all the prognostic factors that we know and love, right? PS2 versus 0, whether or not they're symptomatic, 8 months difference in survival. But just to put it in perspective, where it is that this right and left sit, and I think what we can see is it is a pretty strong prognostic factor that is right up there with, poorly versus moderately differentiated and the presence of metastases at certain sites. We've heard a bit about the predictive, and just to reiterate that there's several studies that have demonstrated that it's predictive for EGFR, and we'll come back to that. Not all do, but certainly the strong predominance is demonstrating that. I thought it was worth showing this slide because I think we've spent a lot of time on first-line therapy, but just remembering that even in this CO17 study, this last line, the gold standard study of best supportive care versus cetuximab, this is what we can see. And the PFS, again, this looks just like the KRAS result, right, where the PFS curve on the bottom left in a right-sided colon really overlapping. Now, this is small numbers now. We've sliced it up now. We're down into 30 per arm. But I think really interesting that suggesting this is a line-independent effect. And then just finally reiterating the 80405 data. All right, so when we talk about this, I think we know that there's this vascular distribution between left and right, but also these differences from embryology. And if you remember back from med school training, for those of you that had to sit through embryology and learn about the patterning genes, the Hox transcription factors, the homeobox genes, I think at some point I'd learned this, but then promptly forgot it. But it's just worth reviewing this again, that there are these patterns that really help define the midgut and the hindgut. And what you can see is that right at that level, left and right, there is a whole different set of transcription factors, these gradients of the homeobox genes that really establish this polarization. And we, of course, use this to describe the midgut and the hindgut. So it is biologically, it's just as much a distinction as between the stomach and the duodenum, is between the left and the right colon when we look at it from an embryologic perspective. So I think it's also worth looking at this from what's the biology of the normal colon, And so I'll spend a little bit of time on this just to reiterate the point that there are certainly different microbiomes. We'll touch on bile acid as one example of differential impact of presence in the colon. We'll talk a little bit about how the colon ages, just like the rest of us. The colon also ages, and then what the premalignant polyps look like. So you've heard a bit about the microbiome, and this is a variety of different microbiomes. There's the microbiome that is in the fecal content. There's the microbiome that is invasive, the fusiform, for example, species that actually invade into the tumor. But then there's this probably most relevant to colon cancer is this microbiome in the biofilms. So biofilms are these kind of mucus layers that sit on top of the colon in certain settings and actually have a fair bit of bacterial content. And what has been seen is that these biofilms are really substantially present more on the right side than on the left. And if you look at that top right figure there, what you can see is that the biofilms are really found at that hepatic flexure and more proximally. When you then start to look at what that means, certainly we're still trying to understand what the biology of this really complex microbiome is. But one of the things that has come up is this recognition that the gut microbiota in the biofilm can be associated with generation of some procarcinogenic agents. One of the ones that's been studied for years, and certainly the gastroenterology field has been very interested in this, is the idea of bile acid. And bile acids are putatively carcinogenic. As we know, they're secreted through the bile, and then they're metabolized and conjugated by the microbiome. So the primary bile acid is actually tenfold higher in the right colon than it is in the left. And what we can see is that there is this process where the secondary bile acids are actually the ones that generate these reactive oxygen species after they've been conjugated by the bacteria that then can cause DNA damage and at least potentially lead to colon cancer. So there's this link, and this is just one of several of links between what the microbiome is doing and what may be happening at the level of the colon epithelium. So the other key difference between right and left is this idea of the field effect. And we're familiar with a field effect of damage in the lungs, for example, from smoking. But there is the same kind of effects can happen in the colon over time. And one of the ways we look at that is through methylation. And that's the idea of this epigenetic regulation of the colon biology. And this is just showing some differences in particular areas of methylation in the normal mucosa. And what you can see here, it's a little small, but the idea that the left colon there in orange, and you can see regions that either have substantially higher or substantially lower degree of methylation. And that this methylation is not stable with time. And so one of the things that happens is that methylation is one of the key components of aging epithelium. And what we know over time is there's some particular areas that are more prone to acquiring methylation over the years. And so this is a very interesting study that's published a few years back. And what this is showing on the right side is MLH1. So this is obviously one gene that we know about, that we know is under epigenetic control and is clinically relevant. And this is looking at that in women and asking the question, over time, what is happening in the normal epithelium? This is not cancer. And this is a cross-section study, but as they look at the colons of normal patients over time, what they can see is a slight increase in this methylation in the sigmoid colon, but a much more dramatic increase in the cecum, right? So as you age, that aging effect is actually more prominent in the cecum. And so I think that is kind of a hint that tells us that our colons age and that our left and right colons don't age at the same way. So the other way to look at what's going on in the normal colon is kind of what sort of precancerous lesions do we see? And we know, of course, the classic chromosomal instability pathway that gives us tubular adenomas, but there's other ones, including what we call the sessile serrated adenoma pathway, which is associated with a lot of this hypermethylation. And we know that this is a lesion that is seen predominantly in the right side of the colon. So it's very different than our classic tubular adenomas, which are more uniformly distributed, or even tubulovillous. You can see one graph here just showing that the cecum ascending hepatic flexure, for example, and even slightly into the transverse, have higher rates of these sessile serrated adenomas with or without the dysplasia. And these, as you know, can have very different patterns. These are two. One is mucus covered, and then one is a very flat adenoma. And so these are ones that we see predominantly in the right side. So this at least sets the framework to say what is it's going on in the normal colon, in the premalignant condition. And so now we probably shouldn't be that surprised that things are very different when we get different cancers that arise out of this right and left side. So we'll go through a few of the things, a few of the phenotypes. So we touched a little bit on this methylation, and I think just worth a quick reminder to say when we talk about methylation, we're talking about an epigenetic effect where there's a methyl group that's put on either promoter regions or regions within the genes that are the intragenic regions that can then modulate gene expression, right? So it's not a mutation or amplification, but it's a very powerful way to modulate the expression of these genes. And cancer obviously uses that a lot. But it turns out that there's some tumors that use this as a major phenotype of the tumor itself. And we use that as a CpG island methylation phenotype, or CIMP, something that's been known for years. The TCGA showed us again here. These are the probes that are methylated, different regions of the genome. And what you can see is there's clearly subgroups that have very distinct levels. And so we'll use this term CIMP-H as a kind of a shorthand for the idea of a hypermethylated phenotype. So when we look at this, what we can see is first just look at the fact that as we go from the rectum to the ascending, we see this methylated tumors going up. And of course, we're familiar with the idea that this right-sided also have higher rates of MSI-H, higher rates of BRAF tumors as well. So it's clear that these kind of key features, these kind of hallmarks of tumor biology that we've known for a while are clearly related to the location in the tumor itself. Now, for the aficionados in the audience, I circled the cecum because I think it's this recognition that it's not just a linear effect from one end, from the rectum to the cecum, but that the cecal cancers actually are starting to actually even look a little different from those ascending colon. And what you can see is a bit of a drop-off there of the CIMP tumors. And it turns out that this has been seen in a few studies. This is actually from our MD Anderson database, but showing that this BRAF, if you look at the red curve, so similar things, just showing that the cecal levels are actually lower BRAF. They peak in that ascending hepatic flexure before dropping off again. But these cecals are a little different, that they have some degree of hypermethylation, but also have this higher rates of KRAS. So if you ask where's the highest rate of KRAS, it's actually in the cecum. In several studies before dropping off in the ascending and hepatic. Minor detail, but for the aficionados in the audience. All right, so we put all this together, what can we say? Well, clearly this idea that age, right, the aging right colon, MSI, BRAF, methylation are all associated with right-sided primaries. When we put that together, these are really strong effects, right? We've known MSI-H 6-fold higher, more likely to be on the right side. BRAF 5-fold, methylated 2.4-fold in some data we presented last year. And we kind of put all this together when these diagrams, what we can see is that the right-sided tumors are really defined by this cluster. And these are metastatic population that we're looking at here, where, yes, that biology is seen in left, but it's a much smaller proportion of the overall phenotype. So another way to look at that is to say, well, there's a lot of different pieces feeding into that. What if we just, instead of looking at what's modulating genes here and what's mutating genes, what if we just looked at the output, right? The gene expression pattern, right? What is actually most relevantly impacting the biology of the cancer. And that's been a complex area, but as has been alluded to already, there's what we call consensus molecular subtypes, where at least the field has come together and said, we think that these are very clear patterns that are coming out of multiple studies. And so we had published this now a year and a half ago, but really defining these 4 different subgroups with an international collaboration. And these have very distinct features, and some of those you can see there that are associated with the mutations and the methylation. And not surprisingly then, what we've shown is that these are associated with very different distributions in the right and the left side. So again, not black and white, but certainly strong associations. You can see the two diagrams there. So the left side really is dominated by CMS1. There's some CMS3 that is higher rates as well there. Conversely, on the left side, we're really looking at either CMS2 or CMS4, right? So this idea that those are the key drivers. Now, again, another little aside is that right and left side don't fully incorporate everything that we may care about clinically. It's a great surrogate, but this is actually some data from a cetuximab monotherapy study that's almost a decade old, but we went back and looked at it and annotated it for CMS2 and 4. As we know, these CMS2 and 4 are really left-sided, so even within the left side, CMS may be associated with differential activity, right? So CMS2 has better outcomes with cetuximab than CMS4, and so I think we're really excited to see some of the data from 80405 that will be presented at ASCO this year. Really ask the question, do these CMS subtypes make a difference? But I think that there is additional information in these gene expressions above and beyond the data from the Sidedness. All right, so one of the other things that we've talked about over the years is this idea of, well, what about Epiregulin and Amphiregulin, right? So this is something that we keep seeing presented because it's compelling biology, but this idea that Amphiregulin and Epiregulin are the highest concentration ligands for EGFR, right? So we talk about EGF, but that actually turns out to be fairly low concentration in human biology. But Amphiregulin, Epiregulin are the dominant ligands. So the idea, well, what if the ligand levels can help predict who could benefit from an EGFR inhibitor? And you may remember a variety of studies. This is, again, studies that went back to that same data set of cetuximab monotherapy and looked at those tumors that had high Epiregulin and high Amphiregulin had better progression-free survival with cetuximab. And it was, I think, other data that said the probability of objective response goes up as your levels of Amphiregulin and Epiregulin go up. And so this is some curves from follow-up studies that had confirmed that. So the question is, well, is it that, how does this biology that we've known about for a bit of time overlap with it? Well, it actually turns out that this Epiregulin and Amphiregulin expression is really higher expression in the left side, right? And that there's very low expression of these genes in the right side of tumors. So again, another alignment of this simple left-right biology with what has been a very compelling and interesting biomarker study over the years. Now, here's where things start to interact. It turns out that these genes are under incredible epigenetic control. And so why are we seeing differences in the left and right? Well, it turns out that this is really related to the fact that all this epigenetic dysregulation going on in the right side of the colon is associated with Amphiregulin and Epiregulin modulation. And so these are now kind of a convergence of our understanding about the epigenetics, the EGFR ligands, and the location of the tumor. So I think we can kind of conclude on the basis of that when we talk about sidedness, that it really is a surrogate for a wide spectrum of known biology. It's as has been termed a kind of the poor man's biomarker that really incorporates all of that. And I think as a field, we have to figure out, do we keep putting efforts into biomarker development, whether it's Amphiregulin, Epiregulin, or do we develop the CMS biomarkers, or do we just say, you know what, left and right, we can get that done pretty quickly. I don't have to wait for the pathologist to give me that result. I don't have to hunt down outside slides. Or do we kind of take left and right as a foundation and then try to better refine within these subclasses? And I think that latter is probably where we're going to be going, but I think we have to start, because of this compelling data, and say, we have to start with our understanding of the left and right and ask the question, how do our biomarkers improve upon this? And that's where we're seeing. So a lot of information I've thrown at you. Apologies for the kind of dump of information, but the idea here is just to make sure that when you see these data, and you'll continue to see more data presented on left and right, that you can at least reflect back and say, yes, this makes sense biologically. This isn't a subgroup ad hoc analysis that we should be that we should be critical of, but really to say it has a fundamental underpinning of biology. So just can conclude that these subtypes have a substantial association with tumor location, and that this happens even in the normal colon, different environment, different genetic pattern, different aging of the normal colonic epithelium, and that I think sidedness really shows these clinically important tumor biology, and I actually think this is going to accelerate our biologically based treatment decisions, right? So I think this is really going to help us better understand what's critical. But it's a really poor man's molecular test, but with a strong biologic foundation. Thank you.